of Strand, presented by Dr. Radhakrishna Bettadapura, a senior software engineer at Strand Life Sciences. I'm Susie Valdez of LabRoots, and I'll be your moderator for today's event. We're excited to bring you this educational web seminar presented by LabRoots, the leading scientific social networking website, and sponsored by Strand Life Sciences. Strand is a leading company in software product development, cutting-edge contract research for pharma, and genetic testing. To learn more about Strand, visit www.strandls.com. Before we begin, I'd like to remind everyone that this presentation is interactive. We want to hear from you, so questions, comments can be submitted via the green Q&A button at the lower left of your screen. We'll answer as many questions as we have time for at the end of our presentation. Also, you are viewing this presentation in a slide window. To enlarge that window, click on the icon in the lower right-hand corner of your slide window. If you cannot hear or see this presentation properly, let us know by clicking on the support button at the top right or the Q&A button to let us know that you're having a problem. Without further ado, let's turn our attention now to the presentation presented by Dr. Radhakrishna Bettadapura. Welcome, sir.
Thank you so much, Susie, for that wonderful introduction. Welcome, everyone, wherever you are in the world, if it's morning or if it's noon or if it's the evening. We're used to making worldwide presentations, so this is one more for us. This presentation is about Strand NGS Server, and I'm going to be talking to you about why clinical bioinformaticians, especially people who do clinical genomics, love Strand so much. A little bit about myself. I'm a senior software engineer at Strand Life Sciences. I have a PhD in computational biology from the University of Texas. I got that PhD in 2012, and I've been working on applied mathematics and bioinformatics and protein flexibility, the nexus of these several interdisciplinary fields for the past 11 years. And at Strand, I actually work on DNA and RNA-seq, and today we'll actually be looking at DNA-seq in some detail. So I promised clinical genomics, and I'm not going to keep you waiting. So here's the PI of our clinical genomics lab. His name is Satish Shankaran, and we're going to be seeing Satish throughout this presentation, and we're going to be seeing him happy. And so the question we want to ask is, why exactly is Satish happy? Right? He's the PI of a clinical genomics lab. So the first thing he has to do, what's the first thing he has to do? What's the first thing that any PI of a clinical genomics lab would want to think about? Building a great team, right? And one of the first questions you ask when you're thinking of building a great team is, how many people do you need? And of course, that usually depends on what you're trying to do. And we're talking about clinical genomics here. And for those of you who may not be familiar with it, all clinical genomics tries to do is attempt to detect certain kinds of genetic diseases. You'll be seeing both these pictures later on in the presentation. For now, just remember that we are trying to detect genetic disease. Right? And so the number of people you're assembling hugely depends on the kinds of genetic disease you want to detect. So in clinical genomics, you typically start with a standard germline panel. And the germline, for those of you who are not familiar, is just the line of inheritance starting from your ancestors and going all the way down to yourself. Right? So it is what you inherit from your parents and your grandparents. And panels that are based on germline diseases, that is, diseases that you inherit from your parents, are called germline panels. And there are some standard panels that are germline. One of them is recommended by the ACMG and contains 25 genes. So these 25 genes are seen to be critical in detecting germline types of diseases. Right? So this is a great starting point for a clinical lab setup. Right? You have 25 genes and you can detect all these different kinds of diseases. Some of them you may already be able to see in the slide. You can see breast cancer 1 and 2, for instance. And there's several other kinds of diseases that this particular germline panel can detect. But this is probably too broad, right? So you want to maybe winnow it down and offer a smaller germline panel for maybe just breast and ovarian cancer, which is actually quite prevalent, not just in the United States, but also here in India, where Strand Life Sciences is primarily based. So hereditary breast and ovarian cancer is actually a huge problem. It's a condition with a lot of sufferers in India. So it's certainly something that you want to be adding a panel for. And that panel is more specific. That is, it has fewer genes than the original ACMG panel. So you see that once you're the PI of a clinical genomics lab, you start with something that's recommended by an organization like the ACMG, and then you start to stratify and start to figure out, that you need more panels that target specific diseases. And this is actually very common. This broadens the scope of any clinical genomics lab. So you generally start by adding something common like breast and ovarian cancer, right? And then you add another thing that's common, another disease, another genetic disease, which is also germline, that's common. And maybe that one is for colorectal cancer, and that comes with its own set of genes or markers, and it comes with its own panel, right? This one has only 14 genes as opposed to the 25 we started with. And pretty soon, you start adding these specific tests, and you end up with several tests, right? One for each family of cancers or each cancer. And this is, again, very typical of the kind of broadening of scope and narrowing of disease specificity that a clinical genomics lab has to go through. So these are sort of exemplary of the kinds of tests that Strand offers in its germline panel, and this is also typical of the kind of thing that you do when you start a clinical genomics lab, right? And this is just the beginning. Right? You have a set of germline tests, but so what? Is that it? Are you done? Remember, you're the PI of a clinical genomics lab, right? Are you done? Well, you're kind of far from done, right? Because you start with the patient, and then you go on to the germline test itself, which you just saw, and you have to do sequencing for that test, DNA sequencing. And then you actually have to do some kind of NGS where you look at the sequenced data with software and interpretation. And then you actually have a physical interpretation stage where people have to additionally understand the kinds of mutations they're looking at and whether they actually correspond to the diseases that are in the test, or whether they're just false positives, for instance. And then once all that is done, you actually have to report your results. Right? So those are sort of the broad stages in clinical genomics, and especially this is sort of the life cycle of a germline test. And we go back to the question about, what about the team now? I mean, how big is it? And where does it start? Where does it end? What do the people do in it? Right? And here's just a simple example. Maybe in the wet lab, you have maybe 7 people. Again, this depends on the number of panels and the size of each panel, the kind of expertise you need. But let's just say ballpark 7. And interpretation and reporting, maybe you want to add another 10. And if you're the PI of a clinical genomics lab, there is something that you definitely do not want to worry about. And what is that? You don't want to worry about the computational stuff, right? You just want the computer stuff that just happen on its own, right? You just want, maybe you want to buy a server or something, or, and somehow offload the work that you have in the software itself, because that really, it should be, according to you, it should be automatic. And, maybe you can add a few bioinformaticians, certainly to look at, the output of the NGS software step, and add a fast server, and maybe you just want to forget about it. And is that enough? That's the question you have to ask again, right? You keep having to ask these questions, and you have to maybe come up with answers, and sometimes it takes a long time to come up with the answers. And this isn't enough either. If it were, I wouldn't be asking the question, right? And there's a critical step here, which is not easy to imagine, but once you start working on clinical panels, it becomes very easy to see in practice. So let me introduce you to 3 members of our team at Strand. On the left is Sanjay, he's from the bioinformatics team. On the right is Aparna, she's from the interpretation team. And in the bottom, you see Pratibha, who's in software, right? So these team members all have to work on different aspects of the particular germline test. So for instance, Sanjay might have to ask himself the question, how do I set up this NGS pipeline? Whereas Aparna from the interpretation team is asking a quite different question. She's asking if a certain variant is legitimate. And Pratibha doesn't have anything to do with either of those guys in a sense, and in another sense, she has everything to do with them, because she's asking a question, how can I make everything easier with a few lines of code or with many lines of code, maybe some scripting. So you see that these team members are all asking different questions, but they kind of need to work together. They need to collaborate. And a lot of their work, a lot of the work of each of these team members depends on the work of the other member. If you were going to draw a graph of dependencies between these teams, and they're all different teams, you're going to see that it's kind of a clique, and that's to the people who studied some computer science and some graph theory before, right? So they're all heavily interconnected. So how do these team members actually work together? How do we get them to work together on the same piece of software? So if we, taking a toy example, if we add 3 more members in bioinformatics, right? And we're asking now the question, well, we also added a server. So we're asking the question, is there a server-based NGS software that supports dozens of users? Clearly at least a dozen in this example, but if you take a practical example with many panels, probably far more than just 1 dozen. Is there a server-based NGS software that supports dozens of users so they can collaborate on different aspects of the same experiment? And the answer is yes, there is. Absolutely there is, and that's called Strand NGS Server. And that's what this talk is about, to make hopefully the viewer understand what the use of Strand NGS Server is and what it does. So the first thing that it enables is what we asked for, which is that clients, it's a server client setup. So clients actually collaborate with each other on the same experiment, right? So it could be just a particular individual with a particular disease that you are trying to analyze and interpret, and it's just that corresponds to one experiment. And all these different clients are working on different aspects of that experiment, just as we wanted. And of course, you have backups, and this is almost a given, with a Strand NGS Server, with the server setup, you want data backups. You just want to make sure that your data is all safe, because this is data, but remember that it's not just any data, it's the patient data, so it's very valuable in many different ways. And you also have a web-based manager, that ensures that anybody with access with a login and password can check in on experiments from anywhere. So these are sort of 3 main features of Strand NGS Server. I'm not going to go into all the features, but these are the 3 main features that enable Satish and his happiness, right? Because now he has Strand NGS Server. So that was as far as the first part of the talk goes. So that's just an introduction to Strand NGS Server. And let's see if we can do something a little more intricate, but right up the alley of anyone setting up a clinical pipeline, right? Which is setting up a clinical pipeline. Right? So remember this germline panel that we saw earlier. Remember that the germline, for those of you who may not know, is it's a series of mutations that you inherit from your parents or your grandparents or your ancestors, It's a germline. This is a germline panel, has 25 genes in it. Every panel you add has its own unique characteristics, right? So for instance, this is a 25 gene panel. It has a certain sequencing coverage. It's based off a certain sequencing technology. It has a number of genes and so on, right? But every panel like this has its own set of characteristics, right? And every panel, therefore, because of its individual nature, requires its own NGS pipeline with its own parameters and their values, right? So a huge part, and I cannot overemphasize this, is just setting up these pipelines. And it takes a long time. So I say day, week, month, year. It never takes a day. It never takes a month. Often it takes months, several months, or even up to a year to just figure out, what are the right parameters for any given panel. And this takes a lot of expertise, a lot of time and effort from various team members, and there are no shortcuts here. And to be clear, that's not what I'm saying Strand NGS Server can do. But what it can do is actually enable you to set up these pipelines in a much easier and more intuitive way, right? So there's, for instance, this is a typical DNA-seq pipeline you see on the top right-hand side. It has alignment, it has realignment, it has variant calling, it has a bunch of other stuff too. This is just the basics. And you have a duplicate removal, you could have base quality recalibration, you could have variant quality recalibration. It goes on and on, right? So there is something that can make this process very easy. That is a pipeline. And a pipeline in Strand NGS Server looks exactly as you might expect a pipeline to look, right? It's a sequence of stages that are connected by arrows, right? So the first stage is connected to the second stage, and the second to the third, and the third to the fourth, so that the logical flow is evident, right? And this is just one of the conveniences. The other convenience that every parameter you see is just right there. You can immediately see it. It's not a problem. If you want to go ahead and change a parameter, you can. If you don't understand what a parameter means, maybe you don't change it, right? I'm just trying to remind some viewers who may not be aware that this is actually a very painstaking process, because if you remember, there are like 12 parameters and there's 15 parameters, and it takes a lot of expertise, but it makes it easier if you just can see everything, especially when you have to do this over and over again, multiple times in order to get things right. It makes sense to expose every parameter like this, and also to be able to add and remove stages as you please. So for instance, here, there are these checkboxes that correspond to every stage that has been chosen. You can uncheck some of these boxes. Note that you can't actually uncheck alignment, because without alignment, you won't have anything, right? But you can uncheck local realignment. You can uncheck some filter stage. You can rearrange so that one of the filter stages follows another, or it can be interspersed with other stages. This automated process might seem simple to mostly everyone viewing this, but the automation is critical if you have to repeat things over and over again, as we've already established that you have to if you want to set up a panel of any size. Any non-trivial panel that you want to set up will require you to do these things again and again. So the whole point of Strand NGS Server is that you can do this very easily, and it enables you to do this. So what else could Satish be possibly worried about, right? So if you have a pipeline, so the pipeline itself, we've established that the setting up of the pipeline is easy. But what about what's in the pipeline? What goes in it, right? So these are the few things that maybe he's worried about. He's obviously worried about the accuracy of, let's say, his variant calls, because usually people are looking at variant calls, right? So he's obviously worried about how accurate his, the various stages that lead up to variant calling are. He's also worried about the speed. He's worried about how much storage is consuming on the server, because remember, every server has a limited amount of storage. So the less you can consume, the more experiments you can get off one server, right? And so on. That's pretty clear. And then you also want to be able to verify your data once you get it. It's not just sufficient to say that something is accurate. You also have to be able to visually sometimes go back and look at things. Right? So these are the 4 things that he could potentially be worried about. So let's just look at them one by one. First of all, the accuracy, and we have a white paper out on Strand NGS and Strand NGS Server, the DNA-seq white paper. We benchmark the accuracy of variant calls. This is, by the way, you see the entire workflow here on the top. What the high accuracy numbers here, the high precision and recall rates, the 99% and so on, are reflective of the accuracy of each stage in the workflow. If one of these stages was bad, then that would reflect poorly on the final variant call set in the VCF, right? Since none of these stages, since every one of these stages is validated, that means that the variant call set that you get at the end also has a very high accuracy. So that's the first step. The second step is, if you want more evidence of accuracy, our in-house clinical lab, remember the one that Satish is the PI of, has 50 clinical team members looking at these variants that are generated from Strand NGS Server on a daily basis. So you have 50 pairs of eyes spending 8 hours a day, every day, for the past 3 years looking at these variants. So that's the kind of validation that you just won't get with most kinds of software. We have an in-house, we eat our own dog food, essentially. That's what we're trying to say. And we're also certified by the College of American Pathologists. We're also certified by the National Accreditation Board here in India as well. So these aspects add a huge deal of weight to Strand NGS Server, and especially its accuracy. You also have the speed. It's twice as fast as the GATK best practices workflow, and that should, again, come as some consolation to clinical genomics, members of clinical genomics labs, especially their PIs. The storage, now, this is another aspect which is very important, because every time you run a stage in DNA-seq, with typical workflows, you're actually incurring an incremental increase in the amount of data that you store, because every file, every additional file doubles essentially what you had before. So it adds on the same size as you had before. So for instance, this in blue is the GATK workflow. As you can see, after sorting, the space that you consume on the disk increases linearly for the GATK workflow, whereas for Strand NGS, it's pretty much constant. And this is because we have great data structures and a logical way of storing things that dramatically reduces the amount of storage that you need for any given workflow. And finally, verification. So we have a lot of deep and intuitive visualizations. We've worked on our visualization for a long time now, again, in collaboration with the clinical genomics team. We've worked long and hard on visualization. So that's one aspect. The other aspect is just the structure. And the structure is exactly what you would expect from a DNA-seq experiment, where alignment is followed by sorting, is followed by some kind of maybe removal of duplicates, or maybe local realignment, and so on. So you see that hierarchy here as you traverse the list. So these aspects actually make verification much, much simpler than they would otherwise have been if you were just looking at a file on a file system. So coming back to our panels, so remember I showed you several germline panels, and we also have somatic panels. So somatic is sort of the opposite or the counterpart of germline. A somatic disease is one that you did not inherit from your parents, but you acquired over your lifespan. So for instance, maybe you stepped out in pollution, or maybe you were exposed to X-rays, or something of that kind, something that occurred to you that caused a mutation to occur in one of the cells in your body. That's a somatic mutation as opposed to a germline mutation, which you inherited. So we have somatic panel, we also have a rare disease panel and also a clinical exome panel. And each of these panels has several sub-panels associated with them. And each sub-panel has a pipeline in Strand NGS Server. And these pipelines are heavily customized, not interchangeable. Each pipeline has been worked on for hundreds of man-hours, and each pipeline has been validated, as you saw earlier, with the CAP and the NABL validation and so on. So why is Satish happy? Because as we saw, these complex pipelines, they aren't a joke. They take years to develop, but Strand NGS Server makes it that much easier to develop. So now we'll look at part 3 of this talk. I'm just going to go over a day in the life of one of our bioinformaticians. And we have several bioinformaticians here, and they're all looking at various aspects of various different workflows, as we saw. And each workflow is tied to some subpanel, and each subpanel is tied to some theme, like germline or somatic or inherited disease. So one common theme that runs through a lot of the things that bioinformaticians do have to do on a daily basis sometimes is to verify the zygosity of an indel. So what does that even mean? What is a zygosity? So all of us have 2 copies of DNA that we inherited, one from our fathers and one from our father and the other from our mother. So our DNA has 2 copies, and a mutation can occur on either one of the 2 copies or both of them. When it occurs on one copy, it's called a heterozygous mutation, and when it occurs on both copies, it's called a homozygous mutation. And some diseases, now coming to diseases, so the whole point of this exercise, if some diseases are recessive, that means that the person is affected only if the variant is homozygous. So the zygosity of the variant is on both copies of the DNA, not just on one, right? And if there's a recessive disease, sometimes you actually have to go back and verify that, that the particular variant is actually homozygous. If it's heterozygous, then it means that the person is not affected, he's just a carrier. So how do you do this in Strand NGS Server? Here is an example of the genome browser view in Strand NGS Server. You can see that there are various details that are being revealed about this particular read list at this particular reference location on the genome, right? So we're on chromosome 1, location about 860,000 or so, right? And what we're looking at is a 4-base insertion at this location, right? And if you just look at this, and this maybe gets a little technical, but stay with me here. This 4-base insertion in purple occurs on roughly half the reads, correct? So since it occurs in roughly half the reads at this location, you might conclude that it only occurs on one of the 2 copies. Remember, we're trying to figure out whether this occurs on one or both copies. But if you actually did realignment here, which is one of the stages in the DNA-seq workflow, you actually see that it's a homozygous variant. So in some scenario where realignment actually was not run, it can be run to verify that a particular indel is homozygous. Another view of this is to look at the variant support view in Strand NGS Server, where you're looking at the same insertion. This is, remember, it's a CCCT, it's a 4-base insertion, right? And this is the variant support view. So the variant support view gives you a few pieces of information. The first one is the cluster ID. So there are 2 clusters of reads here, right? This was before realignment. There are 2 clusters of reads. One that support the reference. So the reference support is indicated by these minus signs. And one that supports the variant, which is in this case the 4-base insertion. And what these figures on the right say is that the first cluster has 17 reads and the second cluster has 15. So that Remember our first assertion about roughly half the reads supporting the insertion? That's what you're seeing here, but in a much more condensed and easy to understand fashion. So 17 out of the 32 support the reference before realignment, but after the realignment, all of them except for one support the insertion, right? So this makes the zygosity of this particular indel, in fact, in this case, it's just an insertion, much clearer. So that was one application and one thing that a bioinformatician might spend some of his time on. The other application of Strand NGS Server, and one that again very commonly used, is in trying to verify chimeric structural variants. And if you don't really know what that means, it really doesn't matter. I'm going to try to explain it to you. This is a portrayal of retinoblastoma in an infant. Retinoblastoma is a form of cancer that invades the eye. It can either be germline or somatic, but we can look especially at the germline case, because the germline case is very illuminating. Let us say, for instance, remember that the germline is the series of mutations that are inherited from either your parents or before them. And let us say one of the parents, let's say the dad, contained a germline mutation that caused retinoblastoma in him as a child. And he's worried about whether his children are prone to it or will get it, right? So he wants to know if he's passed on retinoblastoma through the germline to his children. And this is a very reasonable question to ask, and it's a very reasonable question for which a genetic test could be performed, right? And we also happen to know that retinoblastoma is caused by these long deletions, which are, remember, indels, so deletions are, insertions and deletions together make up indels, and deletions are just one aspect of the indel nexus. So these chimeric structural variants that are responsible for retinoblastoma are actually caused by long deletions. And these reflect in the data as chimeric reads. So here's an example of that. So this is the Elastic genome browser in Strand NGS Server. And what you mean by elastic? Elastic just means that these long deletions, they occur across several hundreds of bases. In this case, if you look at this closely, you can see that this is actually a 9 kilobase deletion. So the Elastic genome browser actually enables the concurrent viewing of distant parts of the genome together at the same point, right? So if you wanted to verify this deletion, it is simply easy to just double click on this Elastic genome browser bookmark, and you will be led to this 9,000 base pair deletion, which happens to occur in the gene responsible for retinoblastoma, hence confirming that this person with this genetic code carries retinoblastoma and will be affected by it in the future, or already is affected by it. So why is Satish happy? And let's just go over the 3 or 4 things that we learned today. So Strand NGS Server is a collaborative server-client model with remote storage, data backups, and web-based data management. It enables the easy setup of several concurrent pipelines, each with its own set of parameters and stages. These pipelines are fast, they are accurate, they are visual, and they are frugal with your storage. And it also contains powerful interrogation tools for bioinformatics validation. And we saw just a couple in the previous slides. So these are some of our Strand clinical panels and the kinds of strengths that we need to support each clinical panel. And of course, to sum, each panel has a sub-panel, and each sub-panel has a pipeline in Strand NGS Server. So remember Satish, we kept seeing Satish, but Satish is only part of the story. He is the PI of our lab team, but we also have so many different members that make this incredibly complex and incredibly varied interdisciplinary field possible. This is just a sampling. These are our the leaders of the bioinformatics team. They serve the bioinformatics team in varying capacities. This is our chief scientific officer and so on. And we also have 50 people in bioinformatics and interpretation. And remember, these are all looking at various aspects of the DNA-seq workflow and various panels therein. And these are our Dr. Chandru is our founder, Dr. Hariharan is also founder, and Thiru is our chief financial officer. So if you need help setting up a server compute infrastructure for especially an NGS platform of any kind, especially a clinical workflow, we can help. We have a great sales team and a great support team. We've had nothing but the best reviews for both of those teams in the past from our various customers. And we hope that if you have any questions about Strand NGS Server, you will contact the numbers here. Thank you so much for tuning in. I'm Radhakrishna or RK. Thank you.
Thank you so much for that informative presentation, Dr. Bettadapura. We want to get right to the questions from our audience. So here's a reminder to the audience members as to how to communicate with us. Questions can be submitted via the Q&A button at the lower left. If you're unable to get to your questions due to time constraints, Dr. Bettadapura will reach out with you via email. So let's take a look at our first set of questions from our audience members. What are the implications of a server-client model? Does the model mean that visualization and computation are separate?
Perfect question. Thank you for that. So I'm just going to go back to the slide over here. Just excuse me for 1 second while I navigate to that slide. So this is just a very simple mock-up of the kinds of interactions that you can have. But what having Strand NGS Server means is that, exactly as you said, client is separate from compute. So the client can visualize while the computation is going on somewhere else, right? So you have the server, you have clients, and then you have compute nodes. And depending on the number of compute nodes available to you, you can have several experiments that are running in the background, and those experiments can go on for as long as they want to, but at the same time, each client is viewing either an experiment that's already done or they're viewing something else. So that's exactly the whole point of Strand NGS Server, is to separate computation from visualization and to separate either of those things from storage. And that's what Strand NGS Server enables.
Looks like we have another set of great questions coming in. Does Strand NGS Server support only DNA sequence, or are there other options or other workflows?
Great question. We support not just DNA-seq, we also support RNA-seq, small RNA, chip-seq, and MEDIT and methyl-seq as well. So there are several workflows that you can use, and sometimes you can be using one workflow while the other one's going on. So we just spoke of DNA-seq in this, but there are other workflows as well. So please contact us for the full details.
Thank you for that. Here's our next question. The part about zygosity is interesting. Could you explain it again?
Sure, zygosity, for sure. Let me just navigate to that slide. Right, so I'm just going to go over what zygosity really means. Once again, you inherit one copy of DNA from your father and the other from your mother. So you have two copies of DNA in every cell of your body, right? And if a mutation occurs, it can occur on one of the copies, so maybe the one you inherited from your dad, or it can occur on both copies. If it occurs on one copy, it's called a heterozygous mutation. If it occurs on both copies, it's called a homozygous mutation. So for instance, you see here in this read list, what's happening is that this purple mutation, it's a mutation because, it's purple because it's a mutation, it's colored purple. Otherwise, normal non-mutated reads are colored gray. So you see that on top, before realignment, you actually have about half the reads supporting the mutation, which might lead one to conclude that this is only occurring on one of the two copies, not both. But if you actually go ahead and do realignment, which you may have neglected to do because maybe you're not setting up, you didn't set up your pipeline with realignment in it, or for some other reason, but if you actually went ahead and did realignment, you can actually verify that the zygosity is on both copies, and it does occur on both, the one you inherited from your dad and the other one you inherited from your mom. So this is very important when it comes to verifying recessive diseases which occur on which are actually the recessive disease is one where the person is affected only if both copies are mutated. So in the cases that we need to know whether two or one copy is mutated, that's when this comes into the picture.
It looks like we have time for one more question. Does Strand NGS Server support base quality recalibration?
Yes, it does. Thank you for that question. In fact, you may have fleetingly seen it in a slide over here. You see that there's a stage called recalibrate base qualities. So yes, it does support recalibration.
Thank you for that. So just as a closing, would you like to take a time and maybe add some last comments to your presentation for everyone, Dr. Bettadapura, before we add?
Sure. Thanks, Susie. First of all, I'd just like to thank LabRoots for enabling this presentation. And we do talk a lot. We have webinars and so on, but this is just another forum, and so it's another opportunity to reach out. I just want to say that for people who are working on especially cancer, I think it's a very exciting time to just be in this field. And we see, especially in developing countries, that the extent to which genetic disease has penetrated, especially India, is unknown. And clinical genomics, it may be a small step, but it is a step in the right direction. And I think it's just a very humbling time to be part of this entire setup. And I think Strand and Strand NGS, server or otherwise, they're just doing a great job of being part of this huge conversation that began maybe 2 decades ago. and is ongoing and needs to penetrate even further if we're going to make any dent in genetic disease. Strand and Strand NGS Server have done a great job so far, and it's a huge it's an honor to be a part of this team.
Thank you so much, Dr. Bettadapura, for your amazing presentation, and thanks the audience member for attending. This has been a wonderful presentation. We want to get close here, and we want to thank LabRoots and our sponsor, Strand Life Sciences, for making today's educational webcast possible. Today's webcast will be available for on-demand until August 2017. Keep an eye out for an email from LabRoots alerting you when this webcast will be available for replay. We encourage you to forward that announcement to your colleagues who may have missed today's live event. Thanks for joining us, and we hope to see you again next time. Bye-bye.